Okay. Hey, step two. Let's go. So, there are even more ancient methods for sequencing DNA, but nobody does them anymore. In fact, a lot of the people that used to know how to do them are actually so old that they're dying off, and we don't have to go back, and this is not a history class. So, we're going to start with kind of the methods that are still in use for sequencing, because I think that's a good place to start. So, we'll start with the good old tried and true Sanger sequencing. This is cheap. You can usually buy vouchers for $5 or $10, and they will sequence somewhere between 500 and 1,000 bases of DNA if you give them the DNA and you give them the primer to use to sequence it. It's really nice and really convenient when you only have a little bit to sequence and you know exactly what, or you know what you're expecting. So for this type of sequencing, you have to already know something about the sequence at the beginning because you have to make a primer. This technique works just like PCR, except we're going to add one extra component that we'll get into in a second. And so just like PCR, you need to know the sequence at this end and at this end. The stuff in the middle can be unknown, but you need a primer that binds here and a primer that binds there, and the only way to do that is to have a pretty good idea of what here and there are. Yeah. So very useful when you've already got some information. Yeah, there are other sequencing techniques, the next ones we're going to get into, that can actually sequence totally random wildcard DNA. Nobody has any idea what it's going to be until the results come out, and those are really cool. Yeah. But tried and true. And yeah, if you start out in the lab, this may be something they have you do. This is like a little training wheels kind of project. So you do PCR. So you have a template that you want to sequence. You have two primers. Actually, yeah, no, you only have one primer. Sorry, one primer, because you don't want to sequence in two directions at once, because that would look like chaos. Yeah. So you have a template. You have one primer. There we go. Now we're better. And in addition to having the nucleotides, the A, C, G, and T, which are deoxy A, C, G, and T, really, yeah, you know and I know, you're going to mix in a little tiny amount, maybe one for every 100 regular nucleotides, of a thing called a dideoxy nucleotide. And so the dideoxy is not just DNA, which is missing an oxygen on the 2' position, but it's dideoxy, so it's missing the 3' hydroxyl group. So it's got no hydroxyls down there. What? It's just got a little hydrogen there. So there we go. They've drawn it. And you're like, cool chemistry lesson. Why do I care? Tell you why you care. You care because unless there is a hydroxyl there, that's where you add the next nucleotide. You're going to get rid of these two phosphates and join that phosphorus onto this oxygen. And if the oxygen's not there, you can't do that reaction. So when you put in one of these guys, these dideoxys, that is called a terminator, because you will not be putting any more nucleotides in the chain. That chain is over. Yeah. It is not going to happen. And so it is stopped. And you're probably thinking, like, all right, it's like PCR, but I only do one primer, so I'm not going to make much of it. And then it's PCR, but I deliberately put in nucleotides that break it? To which I answer, yeah, of course. Yeah, that's how it works. Weirdo. Yeah. Yeah, the other thing that makes it extra special, nice, and fun is that you can attach a fluorescent molecule to this sort of dead-end terminator dideoxynucleotide. And you can attach 4 different ones. So you can have A be a little blue fluorescent thing and C be a little red fluorescent thing or whatever. They usually use T is red, C is blue, G is black. It's like no fluorescence, and A, I think, is normally green, like regular fluorescein. But honestly, it doesn't matter. As long as the sequencing company and or you know what to look for, everything's going to be okay. Yeah, you got to know what you put in the jar, as always. Yeah, so what you're going to do is you're going to run that one primer PCR. And so every time you amplify, you're going to start with the primer. And so you're going to make a product that's exactly the length of the primer plus however many nucleotides you add. And so you know if your primer is, let's say your primer is 20. If you have a PCR product that's 21 long, you know it's your primer plus one nucleotide, and you know it's stopped at the first nucleotide. And so then all you do is you sort out these different fragments by size. And you can do that with electrophoresis. They'll sort themselves out. The smallest ones run faster than the bigger ones. And it's actually so sensitive that you can separate a 21 from a 22 from a 23. And once you've got that, all you have to do is look at the color. Because everything that is 21 long, which is one primer plus one nucleotide, will have stopped at the same nucleotide. And you look at which color that is, and so you'll find out, oh, okay, it's all A's. So that means that first nucleotide after the primer was an A that we just made. Yeah, easy, right? It's going to look like this, and there's a nice little video down at the bottom if you want to go and watch the video. Again, I think by this point in your life, you're pretty good at watching YouTube videos all by yourself, so I'm going to leave that for you. Also, so I don't get a copyright strike or something stupid. But it's going to look like this. You're going to have a little gel, and you're going to run it out, and you illuminate it with a laser. Shoot it with a laser, and it will illuminate all these things. And so you can see the different colors for the different nucleotides. There they are. It's beautiful. Yeah. And yeah, you can read about 500 to 1,000 at a time using this method with these little dideoxys. So it's pretty cool. We tend to call it Sanger sequencing after, I think he's called Fred Sanger, and I think he kind of invented it and was a big deal back in the day. But you know what? Yeah, you can call it dideoxy terminated sequencing as well, but Sanger's a little catchier. Yeah. So yeah, there you go. All right, that's the second part. Let's pause here to regroup, and then let's go for the third part.